Hello and welcome to BioInsights. In our previous lecture, we learned about the architecture of the lipid bilayer of the plasma membrane and decoded the fluid mosaic model. In this video, we will learn about one of the major activities of the cell membrane, that is transport across the cell membrane. One of the most important functions of the plasma membrane is the transport of molecules across it. In this image, we can see a diagram of a lipid bilayer, and if we observe closely, we can see that few molecules are getting transported through it, whereas some of them cannot. For example, oxygen, nitrogen, carbon dioxide, such molecules are getting transported, but smaller ions like hydrogen ion, potassium ion are refused an entry. A large molecule like estrogen is passing through the membrane, but as essential as glucose cannot. This proves that the cell membrane is working or acting like a gatekeeper and is selectively permeable. The membrane is selectively permeable to some molecules present on either side of it. Now let's look at the types of molecules that generally need a pass through the cell membrane. Its permeability depends on three major factors: the size of the substance, the solubility of the substance in the lipid bilayer, and the charge of the substance. So, what are the types of molecules that usually pass through the lipid bilayer? The first type is the small nonpolar molecules, such as gases like oxygen, carbon dioxide, nitrogen. These are smaller in size and they are nonpolar, so they do not usually interact with the lipid bilayer and freely pass across the membrane. The second type is the hydrophobic molecules, like steroid hormones, for example, estrogen, that we used in the previous example. Now we know that these phospholipid molecules, they have hydrophobic tails, and so these hydrophobic molecules such as steroid hormones like estrogen, they are soluble in the lipid bilayer, so they freely pass through the membrane. The third type is small uncharged polar molecules. Though they are polar in nature, they are pretty much smaller in size, such as glycerol water molecules, and also they are uncharged, so they do not usually interact with the lipid bilayer as well, and they also freely pass across the membrane. But in case of large uncharged polar molecules, or in case of ions, the story is different. They are refused a free entry because they are quite large in size. Look at this thing, the large uncharged polar molecules. Though they are uncharged, they are pretty much large in size, like glucose and amino acids. They are larger molecules. And in case of ions, they are charged in nature. Though they are smaller in size, but they are charged molecules. So, how these large uncharged molecules or these ions finally makes it through the membrane bilayer, let's get into that. Various processes involved in the transport of substances across the cell membrane can be categorized as passive transport, active transport, and vesicular transport. So, let's understand these one by one. The first type is passive transport. Many molecules can move briefly across the membrane without any requirement of energy, and this is called the passive transport. It refers to the mechanism of transport along the gradient. Now, it can be concentration gradient or it can be electrochemical gradient. It is also known as downhill movement, that is from higher to lower concentration. Let's understand this with this analogy. Suppose when you are cycling down a slope, it doesn't require much your effort. You move along from higher to the lower elevation and it's natural. Similarly, when substances move from higher to lower concentration, it does not require any extra energy. It does not require any extra energy. Rather, it is energetically favorable. So, when substances move along the gradient, the gradient, the transport occurs without any extra energy expenditure. Such movement is called a downhill movement. Now, there are two types of passive transport that operate at the cell membrane level. This is diffusion and the other one is osmosis. So, what is diffusion? Passive transport of molecules from areas of high to low concentration. It is of two types: simple diffusion and facilitated diffusion. Simple diffusion. Neutral solutes may move across the membrane by the process of simple diffusion along the concentration gradient, that is, from higher concentration to the lower concentration. The rate of diffusion through the lipid bilayer is directly proportional to the solubility of the substances in the lipids. The more soluble it is, the faster it diffuses. Therefore, gases like oxygen, carbon dioxide, nitrogen, these alcohols like glycerol, steroid hormones like estrogen, they diffuse very rapidly across the bilayer. Water may also move across this membrane from higher to the lower concentration, these water molecules. Now, movement of water by diffusion is called osmosis. So, there is a separate name for diffusion in case of water movement, that is called osmosis. Now, what about the transport of ions and polar molecules? Ions and most polar molecules cannot diffuse freely across the lipid bilayer. That's when facilitated diffusion comes into play. Such molecules pass across via the help of various transmembrane proteins which act as transport proteins. So these proteins facilitate the transport of such molecules like ions and polar molecules. So they are called the transport proteins. Now facilitated diffusion is a type of passive transport. So it also occurs along the concentration gradient, that is from higher concentration to the lower concentration, and that is why no ATP or energy expenditure is required. Now, there are generally two types of integral proteins that help in such transport. What are the integral proteins? Why some of them are called transmembrane proteins are all explained in the previous lecture. If you haven't watched it yet, watch it first for better understanding. The link is in the description box. Now, the two types of transport proteins are channel proteins and carrier proteins. What are channel proteins? This is an illustration of the channel protein. Lipid insoluble substances such as polar or charged molecules can pass through the cell membrane due to the presence of channel proteins, which are a type of transmembrane proteins. Now, these protein channels are fixed tube shaped channels. The transportation across or through these channel proteins depends on factors like molecular size, shape, and charge. For example, if this channel is supposed to transport a particular shape or size of molecule, suppose this size of molecules, bigger size molecules like this molecule cannot get pass through this channel. If this channel is destined to pass this shape of molecules, particular shape, a different shape of molecules cannot pass through this channel. Also, it is charge sensitive. For example, if this channel protein is supposed to pass a positively charged ion, for example potassium or sodium, a negatively charged ion like chlorine cannot pass through this type of channel proteins. So this is molecular size sensitive, shape sensitive, and charge sensitive. These channel proteins are highly selective. It permits only one type of substance. Examples of such channel proteins include ion channels, for example, sodium channels, potassium channels, aquaporins, etc. An interesting fact: apart from osmosis, water molecules majorly get transported across the lipid bilayer through specialized protein channels called aquaporins. Despite being a polar molecule, water, due to its smaller size, leaks through the gaps in the membrane, but this movement is very slow in nature. Through aquaporins, water moves way more rapidly along the concentration gradient, especially required in your plant roots or your kidneys. The second type of transporter are the carrier proteins. Large polar molecules cannot diffuse through the protein channels. They diffuse through the help of another type of transmembrane proteins called the carrier protein. This is an illustration of a carrier protein. This type of transport is also known as carrier mediated facilitated diffusion. So, in case of this carrier protein, this is the substance to be transported through this carrier protein. So, when this substance actually binds to this carrier protein, there occurs a conformational change. Now, what is a conformational change? It is a structural alteration in the shape of these protein molecules, and this restructuring actually helps pass this substance across the cell membrane through these carrier proteins. So, carrier proteins can be of three types: uniport, symport, and antiport. What is an uniport? It transports only one type of molecules. So this is an uniport and it transports only one type of molecules. The second one is symport. The transport of one substance is linked with the transport of another substance. So if there is one substance entering or exiting, it is always linked with the transport of another substance in the same direction. So it is called a symport. The third type is antiport. Transport of one substance in exchange of another. So if one substance is entering, the other substance will take an exit through the same carrier protein at the same time. So there is always an exchange. For example, sodium potassium pump. Now these two, symport and antiport, they are called the co-transport system because the transport of one molecule or one substance is always linked to the transport of another substance, be it in the same direction or in the different direction. Let's move on to another type of transport, a major transport process of the cell membrane, that is active transport. A few ions or molecules are transported across the membrane against their concentration gradient from lower to the higher concentration. Such a transport is an energy dependent process in which ATP is utilized and is called active transport. An example is sodium potassium pump. Now imagine you are cycling on an elevated road from lower to higher elevation. It requires more effort because you are going from low to high. Similarly, in case of transport of a substance of or molecule going from lower to the higher concentration, that is against the concentration gradient, and that requires the involvement of energy, generally ATP expenditure. Such type of transport is called an active transport. Since transport occurs against the gradient, it is called uphill movement. We shall see the mechanism of active transport through sodium potassium pump, most extensively studied in the nerve and the muscle cells. Now, before we begin, we must know these are not three different sodium potassium pumps. This is one sodium potassium pump in different stages. Now this is a phospholipid bilayer. This is the intracellular fluid, the cytosolic surface, and this is the extracellular fluid, the outside of the cell membrane. Now if we look at the sodium gradient here, it is low on the intracellular side on the cytosolic surface and it is high on the extracellular fluid, that is outside the cell membrane. Now sodium ions here trying to move against this gradient, that is from low to the higher concentration, from the lower to the higher concentration. Now if we look at this pump, sodium ions here binding to its binding sites. Now as it is moving against this concentration gradient, it requires energy. Now here ATP is being utilized. ATP is getting hydrolyzed and it generates ADP and high energy phosphate. Now this phosphate actually binds to a particular binding site on this pump, and binding of this phosphate to a particular site creates a conformational change in the pump, and that leads to the export of sodium ion, that creates a passage through which sodium ions gets exported into the extracellular fluid. At the same time, what happens? Potassium ions present in the extracellular fluid binds to the active sites on the pump. Now, if we look at the potassium gradient here, it is low on the extracellular fluid and it is high on the cytosolic side. Now, it is also moving against the concentration gradient. So here the potassium ions bind to its binding site on the pump, and that leads to the release of this phosphate group that was earlier bound to this pump. Now the binding of this potassium leads to the release of this phosphate group, and that creates another conformational change, and that brings back the pump into its original shape. So these potassium ions then move inside the cell, and there happens a potassium import, and now the pump is back to its original conformation, and that makes the pump ready for a new cycle. The third type of transport that occurs through the membrane is called the vesicular transport. Is involved in the transport of macromolecules which can neither pass the cell membrane by diffusion nor by active transport. They are of two types, endocytosis and exocytosis. We'll understand them one by one. The first one is endocytosis, which is known as the bulk transport. It involves the formation of vesicles from the cell membrane. It involves formation of vesicles from the cell membrane. Like you can see here, the vesicles are formed from the cell membrane itself. These are of two types, pinocytosis and phagocytosis. What is pinocytosis? It is also called cell drinking. It is the ingestion of liquid material by the plasma membrane. So suppose this is the liquid material and this is ingested by a vesicle. These vesicles are called pinosomes. So such vesicles in case of pinocytosis or cell drinking is called pinosomes. One such example of pinocytosis is reabsorption by the renal tubular epithelial cells. The second type of endocytosis is called phagocytosis or cell eating. It is the ingestion of solid material by plasma membrane. It can be food, it can be debris, it can be some pathogens or foreign particles, etc. These vesicles in case of phagocytosis are called phagosomes. One such example is neutrophils. Neutrophil is one of your white blood cells present in the blood. Neutrophils engulfing bacteria to ensure immunity is an example of phagocytosis or cell eating. The second type of vesicular transport is exocytosis. It is the fusion of vesicles to the cell membrane. See, it is the fusion. This vesicle is getting near the membrane and it is getting fused to it. So it is the fusion of vesicles to the cell membrane. It is reverse of endocytosis. It is also known as cell vomiting. Why? Because egestion, it is the egestion of waste materials or cell products from cell through the membrane. So as the materials or products or waste material, they are getting exported or they are getting outside the cell membrane, it is called cell vomiting. One such example is release of hormones and enzymes by secretory cells in the body. So let's have a quick summary of what we have learned in this lecture from this table. Read along with me and you will not forget anything after this. So transport across the cell membrane are majorly of three types: passive transport, active transport and vesicular transport. Passive transport is of three types: osmosis, simple diffusion and facilitated diffusion. Osmosis is the movement of water along the concentration gradient. Now we will study about the similarity and differences among these three, the simple diffusion, facilitated diffusion and active transport, through this table. Now before that, the vesicular transport is of two types: endocytosis and exocytosis. Endocytosis is further of two types: phagocytosis, that is called cell eating. Phagocytosis is called cell eating, and pinocytosis, that is called cell drinking. Now let's come back to this comparison table. Now there is simple diffusion, there is facilitated diffusion and there is active transport. In case of direction, that is the gradient, in case of simple diffusion, it is along the concentration gradient, that is from high to low concentration. In case of facilitated diffusion, it is also along the concentration gradient, that is from high to low concentration direction. In case of active transport, the direction is reverse, that is against the concentration gradient, that is from the lower concentration to the higher concentration. Now the second topic is energy, if it is required or not. In case of simple diffusion, it is happening from along the concentration gradient, so energy is not required. In case of facilitated diffusion, it is also a type of passive transport, high to low in direction, so it there also energy is not required. But in case of active transport, as it is happening against the concentration gradient, energy is required, ATP is utilized. If protein is required, no. In case of simple diffusion, protein is not required. But in case of facilitated diffusion, protein is required. We have studied about the channel proteins and carrier proteins. In case of active transport, protein is also required. It can use carrier proteins. If it is specific or not, in case of simple diffusion, no, it is not specific. In case of facilitated diffusion, it is specific. We have studied about the specificity of channel proteins, carrier proteins, etc. In case of active transport also, it is highly specific in nature. So this is it for this video. In the next video, we will study about the cell wall that is exclusively present in plant cells, and we will also study about the cell envelopes. We will also study about the cell envelopes, that is a distinct feature of the bacterial cells or prokaryotic cells, especially the bacterial cells. So till then, study hard, take care, and thank you.